Hey everyone, good to have you here. You may have heard of him as the man who will make you live forever, or at least until you get hit by a car. But here he is, Aubrey the Grey.
Thank you, Shanee. I like the way that Shanee introduces me. It's great. I've had the good fortune to be introduced by her twice today. All right, so thank you for coming. I'm going to try and tell you all about what we are doing at Sense Research Foundation. I have already made my first mistake, which is to leave the clicker on my chair. Right. So we are, as Shanee said, we are interested in stopping you from getting sick as you get old. It has been a source of frustration to me that we get sick as we get old for quite some time, and an even greater source of frustration that it isn't a source of frustration for everybody. So I'm trying to change that, but also, of course, actually change the facts of the matter to try to actually deliver medicines that will allow us to stay healthy. And so I'm going to spend the next hour telling you all about how. First of all, the success story. So this is simply, for illustration, statistics from the USA showing the proportion of the population that are over the age of 65, which was way down at 8% or so in 1950, and is projected to be up around 22% in 2050, which is pretty dramatic. And of course, this is a cause for celebration because it is the consequence of a great deal of success over the past 100 or 150 years in bearing down on the diseases that used historically to kill an awful lot of people in infancy or in childbirth and so on. And the result is that pretty much everybody is surviving long enough to reach the age of 65 or more. But it also has, of course, created some problems. The economic problems are perhaps foremost in many people's minds right now. And again, here I'm just showing US statistics for illustration, but of course it's the same throughout the industrialized world. We have this problem that even though the economy has been growing nice and fast, I'm going to use this one where you can actually see my pointer, all right? I've never understood why these screens can't be made to actually reflect laser pointer light, even whatever. So yes, so this is the US economy, the red line, going up at a nice steady rate. And if you look at the rate of medical expenditure in the US over the same time period, which is the last 50 years or so, then it looks perfectly manageable, really. It doesn't look particularly scary, but it looks a great deal scarier if you take the trouble to divide one by the other, which is what you get, which is where the green line comes from. And the green line shows that obviously then, therefore, the proportion of the GDP that is being spent on medical care, and it's gone up from 5% 50 years ago to 18%, which is extremely scary, and of course is the reason why we have all these concerns about pension funds running out of money and Medicare running out of money and so on. So the question is, first of all, why? Why have we got this situation in the first place? Putting it in starker terms, why have the diseases and disabilities of old age, which of course dominate medical expenditure today, why have they been so much more resistant to medical intervention than the infectious diseases that we have more or less entirely eliminated over the past century or so? This is actually pretty paradoxical when you think about it. And in order to answer this question, the first thing I think we need to do is to ask ourselves, what is aging anyway? What is the ultimate substrate for these age-related diseases? So I'm going to give you a definition of aging now that I'm going to be using for the rest of the talk. And this definition is specifically geared to trying to eliminate the distractions that often come when aging is discussed. There are many definitions of aging out there that are really do more harm than good because they just confuse the issue. But this one brings it down to its essentials, and I think it's useful. Essentially, what I'm trying to do here is to emphasize that the aging of living organisms is really no different, at the bottom line, from aging of inanimate objects, like cars or aeroplanes or whatever. It is simply a fact of physics, never mind biology, that machines with moving parts do themselves damage as a side effect of their normal operation. And it's also a fact of physics that that damage accumulates and therefore, eventually, it exceeds the level that the machine is set up to tolerate. So that's true for a car, it's true for an aeroplane, and it's true for a human being. It's true for the human body. The human body does a lot of different types of damage to itself, which I'll be getting onto over the next while. And therefore, we are subject to a whole bunch of different eventual failure modes, so to speak, of the body. And that's really all it's about. So really what that means is that we must address a very fundamental misconception that is prevalent within society today concerning the diseases of old age on which we spend so much money. Diseases like Alzheimer's and most cancers and cardiovascular disease and type 2 diabetes and osteoarthritis and so on and so forth. These diseases are normally viewed as diseases. In other words, they are viewed as in some sense similar to infections and potentially amenable to the same kind of medical interventions that infections are amenable to. But they're not. They're actually all part and parcel of one single process, the accumulation of damage that the body does to itself in the course of its normal operation. So, not only are they, as we all know, widespread and staggeringly costly, but they are universal. The only way that you will not get a particular disease of aging is if you die beforehand of something else. You are going to get it. And furthermore, they are not medically curable in the strict sense of the word curable. What do I mean by the strict sense? I simply mean that we cannot eliminate them from the body in the way that we can eliminate an infection. If you eliminate an infection, then the person isn't going to suffer from that infection again unless they get reinfected. But you can't do that with things that are side effects of being alive in the first place, other than by eliminating being alive in the first place, which would kind of defeat the object. So let's not do that. But, of course, that means that does not necessarily mean that we can't address the diseases of old age using medicine. It just means that we have to rethink our starting point in designing the medical interventions that we might use. I'd like to put it this way, even more starkly. Most people think of the various causes of ill health along the lines that I'm showing in this table. There are various types of disease. There's infections, communicable diseases, and there are congenital diseases that small proportion of us have because we got dodgy DNA of one sort or another. And then there are the chronic age-related diseases, which are the subject of so much concern and so much expenditure today. And then, in most people's minds, there is this completely separate category called aging itself, which is composed of these non-specific, rather poorly defined things like sarcopenia, which is the decline of muscle mass in the elderly, or gain of fat mass, or decline in function of the immune system. These things that we don't really think of as diseases. That's what most people have in their minds when it comes to ill health. But it's nonsense. This is the way that we ought to be thinking about this. All the columns are the same here, it's just where the black line is that's changed. There are diseases, and then there's aging, but the diseases of old age are part of aging. The only difference between column 3 and column 4 here is that column 3 consists of those aspects of aging that we have taken the trouble to give disease-like names to. It's purely semantic and no more. That is an absolutely fundamental thing that I'm going to be leaning on for the rest of the talk. So I wanted to make sure that you have it in your heads very clearly at the outset. Now, what does this mean in economic terms? I mean, we can look at it in many terms, humanitarian, of course, but let's just do it in terms of economics. And here, for the moment, I'm going to move away from medical expenditure to the expenditure on medical research, because unfortunately it's the same story, perhaps in even starker terms. And again, I'm going to use US statistics just for illustration because I happen to have them. But again, this is absolutely the same across the industrialized world. So the NIH, the National Institute of Health, is the counterpart of the Medical Research Council in the UK. It's the government body that is responsible for the essentially all public expenditure on medical research. And it has a nice healthy budget of $30 billion a year. But only 3% of that budget is spent on aging, the National Institute on Aging. That is pretty bad news, given that the vast majority of medical expenditure and, of course, suffering caused by disease and so on in the US is driven by aging. It doesn't make sense. but that's how it is. That's bad, but it's not nearly as bad as it gets. Even within the National Institute on Aging, only about 1/6 of that budget, that $1 billion, is spent on understanding the biology of aging. The rest goes on specific research on Alzheimer's disease or geriatric medicine, or even the social gerontology, studying how to preserve the dignity of the elderly, things like that. Which is all very well, very important stuff, but it's not what would actually give the bang for the buck. So it's a disgrace that it's so small. And that's not the end of the story. The fact is that even within the Division of Aging Biology of the NIA, or the NIH, the amount that's actually spent on doing something about aging is way under 10%. That number 10 million I've got there is a very generous estimate. You know, really, that's amazing. Most gerontology consists of, it's a bit like seismology. You know, people who study earthquakes, they understand that what they study is bad for you, but they're not proposing to actually do anything about it. It's all about just getting out of the way, really. So you know, it's a bit sad. And I mean, to put it in real perspective, what we're talking about here is that my little foundation, the Sense Research Foundation that's been created around my work, which has only existed for a few years, and we only have a budget of $4 or $5 million a year, it's on the same order as the US government. That is fairly shocking. So something better be done, really, and that's why I go around the world giving talks like this. This is essentially where we are today. If you look at the bottom of this diagram, what I'm saying is what I told you already. Metabolism causes damage, causes pathology. In other words, the network of processes that keep us alive from one day to the next, which is what biologists call metabolism, causes various types of molecular and cellular change to the composition and structure of the body, which I'm calling damage. And that damage accumulates, and eventually, when it exceeds a certain threshold, it impacts the function of the body, and that's when the diseases and disabilities of old age begin to emerge and to progress. And all of what we try to do today to attenuate and alleviate the pathologies of old age consists of what I'm calling here the geriatrics approach, geriatric medicine. It means, in a nutshell, completely ignoring everything that I've told you in the last 5 minutes. It means pretending that the diseases of old age are just like infections and can be eliminated from the body. It's obviously complete nonsense. First of all, for the reason I've told you, namely the damage is continuing to accumulate, so obviously anything that comes under the geriatrics heading is going to become progressively less effective as time goes on, as the damage becomes more and more irresistible. And secondly, because the pathologies of old age are quite numerous. You're not supposed to be able to read this slide, don't worry. This is simply a nice selection of the things that go wrong with us as we get older and that we would rather didn't. So it's completely hopeless, but we spend a spectacular amount of money on it. Now, I am by no means the first person to point this out. For the past 100 years, there have been a few people out there trying to get people to understand that actually if we want to do anything about aging, we have to intervene at an earlier stage in the chain of events that I've just shown you. And that's what has given rise to what I'm calling here the gerontology approach, which is the approach favoured and pursued by that rare breed of people who study the biology of aging with a view to actually doing something about it in due course. However, that approach has also failed completely to deliver any kind of actual impact on the ill health of old age. Why should it be? Well, what is the gerontology approach? It consists of essentially trying to clean up metabolism, trying to slow down the rate at which metabolism creates damage. And that sounds like a great idea, because certainly if we could do that, it would postpone the age at which the damage reaches a pathogenic quantity. But there's a couple of problems with the gerontology approach too. First problem is that it's only slowing down the accumulation of damage, so that means that the later you start the therapy, the less benefit you're going to see. But in other words, the less time you're going to have to extend before the pathogenic threshold is reached. And that's a bit of a shame, because some of us may already have the misfortune to be in middle age or older by the time that the therapies in question are even developed. So that's a bit of a shame. But worse than that, the gerontology approach suffers a problem that is rather analogous to this problem, namely, hello, there we go, this problem. metabolism is rather complicated. This here is a simplified diagram of a small subset of what we know about how metabolism really works, about how the body works. And as you can see, it's a bit hairy. And as engineers, or as people who work with engineers, I'm sure you all understand that it's pretty implausible that we would be able to, in some way, tweak this vast network of uncommented spaghetti code in a manner that would stop it from doing the thing we don't want it to do, namely creating damage, without also stopping it from doing stuff that we do want it to do, like keeping us alive. So, it's a waste of time, really. There's no way in the world that we're going to actually make this work. But if that were the end of my story, I wouldn't be standing here. You knew that. So what's the get out? How do we extract ourselves from this? Well, ultimately what we do is we go back to the analogy that I started with some minutes ago. This car is more than 100 years old. And, of course, there aren't many cars like this, but they're not all that rare. The question I want to highlight, the point I want to highlight about this car is that it was not designed to last 100 years. It was designed to last maybe 10 or 15 years before it would fall apart and you go and get a new one. And the reason it's lasted so long is understood. We all know that the way that enthusiasts allow their cars to live a long time is by doing really comprehensive preventative maintenance on them, by simply repairing the damage that the car does to itself as a side effect of its normal operation, a phrase that you may recognize from a few minutes ago. Now, that means that we can realistically ask ourselves, well, maybe we could actually do the same thing to the human body. Maybe we could engage in preventative maintenance, and if it was sufficiently comprehensive, maybe it would have the same effect. It would greatly extend the healthy longevity of the machine that we call the human body beyond the longevity that it was initially set up to be able to achieve. This is what that means in terms of comparing and contrasting with the other approaches. Rather than trying to slow down the rate at which metabolism creates damage, as the gerontology approach tries to do, or indeed the rate at which damage creates pathology, as the geriatric approach tries to do, instead, the proposal is to uncouple those two processes from each other, to actually go in and periodically repair the damage so that the damage, even though it's still being created, never actually reaches the pathogenic threshold. And what I'm going to be trying to persuade you of today, in the rest of the talk, is that this approach is very much more feasible than the other two. That it has a very realistic chance of, we have a realistic chance of pulling it off within the foreseeable future. So in order to get onto that, I'm going to now start talking about actual biology. I'm not going to get too technical, obviously, but I'm going to try to give you a feel for what this is all about. The first thing we have to do is to define the problem in concrete terms, in down-to-earth, actual biological terms. And that is what this table does. Here we have a list of 7 types of damage, 7 categories of damage. And as you can see, each of these things is a genuine, bona fide physical, biological thing. Cell loss simply means cells dying and not being automatically replaced by the division of other cells. Very simple concept, right? And this is a classification which I have been using for this purpose for quite some time. Now, you may have a couple of questions about this classification, and I want to answer them right at the outset. The first question you may have is, well, hang on, what's so useful about this classification? You recognize, as I've already in fact highlighted, that there's an awful lot of things that happen during aging. There are indeed an awful lot of different types of damage that go on. So, 1,000 things can be classified, can be grouped into 7 different categories in many different ways. What's so useful about this one, you may be asking yourselves? The answer is that within each of these categories, there is a particular generic type of intervention that exemplifies, that illustrates the maintenance approach. Within each category, there may be many examples, but all of them can be, in principle anyway, addressed by, broadly speaking, the same therapy, differing only in details from one example to another. That's the absolutely fundamental thing that you're looking for in any kind of classification of a problem into subproblems. So that's what this classification is all about. The second question you may have is, well, hang on, how do we know that this is exhaustive? How do we know that there isn't an eighth category and a ninth one? And of course, we can't absolutely know. But the good news is that we have at least a good circumstantial argument that, that it probably is an exhaustive classification. What is that circumstantial argument? Here it is. It's been the same classification for more than 30 years. All of these things have been major topics of research and discussion within gerontology since at least the early 1980s. Now, one could say, well, that's not really a fair argument because people weren't really looking to classify damage in this way back then. And that's kind of true. But it's increasingly not true because the fact is I've been out there making a nuisance of myself and challenging people to actually come up with other categories to add to this list for more than a decade now, and I do seem to be getting away with it. I am, like, getting away with it. So that's pretty good news. I mean, it's a circumstantial argument, but it's quite compelling, really. Now, I'm going to spend a few minutes now highlighting the relationship between damage and pathology. And this is really important because ultimately it is, it's where the rubber hits the road. At the end of the day, we need to actually convince ourselves that if we could repair all of these types of damage, we genuinely would have this effect on the various pathologies of old age. And what I want to emphasize to you here is that the relationship between damage and pathology is in some cases quite complex, but it is established. It is well known. The stuff I'm going to be talking about in this particular section for the next couple of minutes is not innovative ideas that I've put forward or that Sense Research Foundation has put forward. These are things that no one working in these fields would dispute. So let's start with cancer. Cancer is the simplest example, because in this case, it's pretty much a one-to-one relationship between the pathology and the damage. There's this one category of damage that I define as division-obsessed cells. In other words, having too many cells because there are cells which are dividing when they're not supposed to. That's pretty much the definition of cancer. So it's a nice, straightforward one-to-one relationship between a type of damage and a type of age-related disease. But in general, it's not quite like that. So the heart is a good example. The heart is an organ that can go wrong in a whole bunch of different ways in old age. And it turns out that the different ways it can go wrong are driven by different types of damage. So let's first of all take atherosclerosis, the accumulation of fatty deposits in the major arteries, which leads, of course, to heart attacks and strokes, and therefore to the major killer, to the number one killer of the Western world. That comes down, at the end of the day, to this thing here I'm calling intracellular junk, molecular garbage that accumulates within cells. In particular, what happens here is that there are a type of white blood cell called a macrophage, which goes into the artery wall for the purpose of clearing up detritus, and it's very good at it, but the macrophage gets poisoned by contaminants in that detritus, which eventually cause it to become more of a problem than a solution. And I will come back to that at some length later on, because we've done some quite important work in addressing that over the past few years. The second type of way that the heart can go wrong during old age is arteriosclerosis, which is the stiffening of the major arteries. This is something that is very important in terms of pathology because it leads to increased blood pressure in the elderly and therefore to things like kidney failure. And the ultimate molecular driver of arteriosclerosis is this thing at the bottom in pink, the stiffening of this lattice of proteins called the extracellular matrix, which essentially gives each of our organs the shape and physical properties that it has. And in particular, the extracellular matrix of the major arteries is extremely essential in terms of giving them the elasticity that they need to dampen the pulsation of the heartbeat, and thereby protect the more fragile capillaries and smaller parts of the circulation from the blood pressure. So that's arteriosclerosis. The extracellular matrix becomes less elastic as a result of chemical modifications, and we need to fix that. Then there's amyloidosis, which is actually has been discovered quite recently to be a major killer of the really elderly. People who live more than 105, 110, it turns out that most of them, or certainly half of them, die of a disease called senile cardiac amyloidosis, which is essentially the accumulation of molecular garbage outside the cell, that thing I've got in blue here. That's all about a type of protein that gets into this kind of fibers that essentially weaken the joints between the various muscle cells that make the heart up and weaken its ability to beat. So, so that's rather bad for you eventually, of course. And then finally, there is cell loss. The heart actually does not beat on its own. It only beats when the brain tells it to. And the cells in the heart that mediate that signal, they're called pacemaker cells, and it turns out that they don't maintain their numbers during age. They die progressively, and eventually you haven't got enough of them, and the heart stops listening to the brain, and it just doesn't beat anymore. So that's another way that the heart can stop. So, as you can see, the relationship here between damage and this particular type of pathology is quite a complicated one, a many-to-one relationship for sure, but, like I said earlier, it's really well understood, and that's the important thing I want to get across. Same with Alzheimer's disease. Alzheimer's disease was defined about 100 years ago or more as the combination of these two things here, a type of junk inside neurons called tangles and another type outside called plaques. And now, of course, we also know that there's a lot of cell death in Alzheimer's disease. So again, a complex relationship, but a well-understood one. Big thing to take into account here and to emphasize is that these non-specific aspects of aging that we don't give disease-like names to, the same applies. Most of these things, pretty much everything except division-obsessed cells, ultimately can be linked causally to various types of decline in function, like loss of muscle or loss of immune function and such like. So this is really good news. We have a very clear idea of this linkage. Right then, enough about the problem. Let's go to the solution. So, what is the preventative maintenance approach? It falls into these four types of approach: replace stuff that's gone missing, remove stuff that has accumulated that you don't want, sometimes in situ repair of stuff that you don't want to remove or to replace, and sometimes reinforcement. That means essentially breaking the link between damage and pathology, somehow doing something to the body that means that the damage can still accumulate, but it no longer actually has any pathogenic effects. And when we come down to specifics, it's all about this. So I won't go through this in detail. I'm going to touch on a couple of the examples here in a moment to look at the details of the pathologies. But essentially what I'm showing here is that we know exactly how to do this in principle. Some of it is much more than in principle. Let's take cell loss. The main way, the generic way to restore cellularity, cell number, to a given tissue is, of course, stem cell therapy. That's true for cell loss during aging, just as it is in terms of any kind of acute trauma or injury for which stem cell therapy may originally have been developed. Parkinson's disease is a great example of this. It's a disease in which cells in one particular part of the brain die unusually rapidly, and stem cell therapies for Parkinson's, they were first attempted nearly 20 years ago when we really didn't know what we were doing with stem cells. But even then, they sometimes worked really, really well. And now there's a great deal of resurgence of interest in this and new clinical trials going on. And I think there's quite a good chance, at least 50%, that within as little as 10 years from now, we will be able to say that Parkinson's disease has been cured. So I won't go down the others now because I'm going to touch on some of them in detail. What I'm going to do is talk about how far advanced they are. And I'm going to start with the ones that are most advanced. So I've just mentioned clinical trials for stem cell therapies are already in progress in certain cases, and that's great, that's wonderful. And actually, it's the main reason why Sense Research Foundation doesn't do anything to speak of, hardly anything in that whole area, because our contribution would be a drop in the ocean. Another case that I want to highlight in a little more detail consists of this business of molecular garbage outside the cell, which in general comes down to this thing called amyloid. Amyloid, as I mentioned earlier, accumulates in the heart, and it also accumulates in the brain during Alzheimer's. That's what senile plaques are. And 15 years ago or so, it was determined, at least in mice, that one could get rid of this stuff just by immunization. Essentially, one could trigger the immune system to engulf the material, get it inside the cell, inside phagocytic cells, and this was enough to get rid of it, to remove it, because the machinery for breaking things down inside the cell is much more powerful than what exists outside. And this moved, in the case of Alzheimer's disease, very rapidly to clinical trials. And those clinical trials got to phase 3 and reported just a year ago. And the reaction was frustrating. The trials did not achieve their clinical endpoints. That isn't what was frustrating. I would have been absolutely amazed if people given these treatments to get rid of plaques had actually exhibited any significant restoration of cognitive function. Why? Because it's not the whole of Alzheimer's, dummy. I mean, Alzheimer's is these 3 things that happen. It's not just amyloid accumulating, it's also tangles accumulating inside the cell, which was not targeted by the therapy, and so there was no reason why we would expect them to go away. And similarly, the cells that had gone missing, that had died, were not being replaced. There's no clear chain of causation. It's not as if we already had any reason to believe that if you removed plaques, then the tangles would go away on their own, or the cells would somehow regenerate. You know, I think it's a bit like what I'm saying at the bottom there is, it's a bit like saying, you know, let's take a car apart and put the individual components out in a line, and then let's pour burning petrol over them, and we'll expect to see some kind of motion. You know, it wouldn't happen really. You know, so it's frustrating that medical researchers and scientists are often rather careless in their evaluation of how technology is supposed to work, especially divide and conquer technology of this kind. Let me talk about another case, and here I'm going to emphasize the role of the private sector, and especially the role of venture capital, because obviously in the IT world you guys are quite tuned into all of that, and you may be wondering already, you know, why does a non-profit such as Sense Research Foundation really need to exist if all of this is so clear? So I'm going to talk about this case because it's an interesting kind of intermediate transitional case. It concerns death-resistant cells, which is a category that I've defined that simply means the other way of having too many cells. You can have too many cells because cells are dividing when they're not supposed to. That's what cancer is, as I mentioned a moment ago. Or you can have too many cells because cells are not dying when they are supposed to. People often overlook that because they think, well, cells are not supposed to die, right? But that's not true. It turns out that there are certain parts of the body in which cells are absolutely needed to die, required to die in order for the whole system to function. So we'd like to be able to get rid of these things. And for a long time now, I've been saying that the way to do this is with something called suicide gene therapy, where you use engineered viruses to introduce a toxic gene into cells. This gene creates a protein that will actually kill the cell. And the way you do it is by ensuring that the protein is only synthesized within the cell in the case where the cell has got into this bad state where you'd like to get rid of it. So this is something that is routine in the lab, in mouse experiments, but it's very dangerous, and so it hasn't got to prime time for the clinic yet. But it will. So that was the thing I was interested in making happen. And the good news is that a couple of years ago, a group at a very prestigious institution called the Mayo Clinic actually had a go at pretty much that. What they did was, since they were working in mice, they didn't actually need to resort to this engineered virus business of suicide gene therapy. They just introduced the suicide gene transgenically, so that it was in every cell in the mouse from the time the mouse was conceived. And what they did was they made a combination of 2 things. First of all, they introduced a second mutation into these mice, a different mutation, which caused these death-resistant cells to accumulate much, much more rapidly than they normally would, but which did not cause any other aspect of aging to be accelerated. So these mice basically died of having too many death-resistant cells. And they died pretty quickly. They died at less than half the normal age. This mouse at the bottom here is such a mouse. It's less than a year old, and it's only got about a month to live if it's lucky. And as you can see, it's very unhealthy already. So what they did was, this research group, they engineered this suicide gene into these mice, and they engineered it in what was called a drug-inducible way. In other words, in such a manner that they could activate that gene, but only, of course, in the cells that they wanted to kill, by giving the mice some simple drug in their drinking water. Until that time, even the bad cells would not actually die because they would not express the suicide gene. All right, and so what they did was they waited until, let's say, halfway through these mice's lifespan before giving them the drug and activating the suicide gene, so that a whole bunch of these cells would already be in existence. And the result was very nice. The top mouse here is one like that, same age as the bottom one, and as you can see, it's perfectly healthy, doing very fine. And they measured the relative health of these mice in all manner of different ways. This graph on the right is just one of the dozen things they did. It's to do with muscle mass. But anyway, so this was all very good news. Now, here's the interesting thing. They did this in an academic institution, but they decided to go out and start a startup company, and they got VC money really pretty fast. Why is that interesting? The reason it's interesting is because the experiment was extraordinarily preliminary. First of all, it was in mice, and we all know that stuff that's in mice often takes quite a long time to translate to the clinic. Second of all, they were working in this artificial system where they had accelerated one aspect of aging very, very dramatically. So there's no evidence from this experiment showing that the senescent cells, these death-resistant cells, are actually all that bad for you in a normal mouse, in a normal lifespan. So, this was really, a bit of a leap of faith. Third thing that was very preliminary about this experiment was that it was a transgenic experiment where they introduced this gene into the mice that allowed them to kill the relevant cells at the point of their choosing. Now, that tells us absolutely nothing about what the actual plausibility is of the company's business plan, which is to create an actual pharmaceutical, a small molecule drug that would do the same thing. There's absolutely no way that there's any evidence from this experiment that that is even possible. And the fourth thing is that even if you forget all this and you just presume the investors were stupid, or that at least they thought that their eventual customers would be stupid, that they might be going for some kind of very superficial bottom line, that bottom line would be longevity. There's one thing I haven't told you about this experiment so far. You probably think that these mice at the top, right, probably lived a normal life, like 2.5 years, as against only a year for the mouse at the bottom. That turns out not to be true. It's actually the case that the mice at the top died after a year, same as the mice at the bottom, because it turned out that there was something else that the gene did, that the mutation did, that caused the accelerated accumulation of death-resistant cells, that was not to do with death-resistant cells. It just basically made the heart unhappy, and so they died anyway. So on every single measure that you could think of, this is a massively preliminary experiment. But they got their money. How did they do it? I think that the answer is a very encouraging answer, namely that investors, smart investors, who are looking at things to do with the eventual delivery and development of anti-aging medicine that actually works, have been paying attention to the numbers. They've started paying attention to the fact that the real anti-aging industry to come, the anti-aging industry consisting of therapies that work, is going to be the biggest industry ever by a very large margin. It's going to be the industry of all time. So that, of course, means that if you want to get into it, if you want to make money, then your calculations of decision about whether to go for it or not will use different numbers. You will be willing to accept higher risk. You will be willing to accept a longer time frame before exit, all those things, right? That seems to be happening now. That's why this kind of thing is actually occurring, and it's very heartening indeed. All right, so you may think, well, okay, that's great. So even something really preliminary is getting funded by the private sector. Why does Sense Research Foundation need to exist as a non-profit funded by philanthropy? Well, I'm afraid the answer is that even though some things are moving forward nicely like this and are in the position of being able to get money out of venture capital, a lot of things that need to be done are still at an earlier stage than that. Early enough that I don't think the calculations are going to fly for a while. The mission statement of Sense Research Foundation is that we want to create a rejuvenation biotechnology industry. Don't get me wrong. We definitely want that to happen. But there are some things where it's not ready for prime time yet in the private sector. And I'm going to give you an example here because it's a good example of the stuff that we've been doing. It's showing very good proof of concept, but it still has a little way to go. And it's to do with this thing, molecular garbage inside the cell, and it comes back to the number one killer in the Western world, atherosclerosis. Atherosclerosis, as I mentioned earlier, starts with white blood cells, a particular type of white blood cell called a macrophage, going into the artery wall and clearing up garbage, clearing up detritus. And it's very good at it. It basically recycles various materials and exports them back out for reuse. But there are certain contaminants in that detritus which the macrophage is not equipped to handle. And those contaminants poison the macrophage, and eventually it doesn't work. The particular part of the macrophage that stops working is this thing called the lysosome, which is kind of the garbage disposal machinery of the cell, and it's a really important part. And so when the lysosome stops working, the cell stops being able to do things it used to be able to do, and eventually it turns into this kind of undead thing called a foam cell, which is what we have showing here. This is the first visible stage in atherosclerosis. And actually all of us have foam cells in our major arteries, even as young kids. But we don't have very many of them, and when we don't have very many of them, lo and behold, they're not harmful. But eventually there's too many, and the environment, the other cells around, start to get a little angry. They get inflamed, and bad things start happening. More white blood cells pile in an attempt to solve the problem, but they get poisoned as well, so they become part of the problem, and the plaque grows and grows, and eventually you end up with the plaque bursting, and that's when you get heart attacks and strokes. So, what could we do about this? Well, about 15 years ago, I proposed that what we really need to do to stop this kind of thing happening, and a similar kind of thing happens with different toxic molecules in, for example, macular degeneration, the number one cause of blindness in the elderly. The thing we need to do is simply equip the relevant cells that are being poisoned with additional machinery for breaking things down, so that in particular they can break down the things that are poisoning them. And it turns out that that's not so hard as all that. In the case of atherosclerosis, there is just one particular compound, it's called 7-ketocholesterol, which appears to be responsible for at least most of the problem. It's not by any means the only thing that accumulates in macrophages over time, but it does seem to be the most important in terms of its abundance and its toxicity. And what we decided to do was to take a leaf out of the book of environmental decontamination, not even a biomedical field at all. So, of course, no one working in gerontology knew anything about this. But what we did was we simply used the idea that you might be able to find bacteria in the environment, in the soil, that could break down the material that is poisoning these macrophages, this compound 7-ketocholesterol. Now, our idea was not, I should emphasize, that having found such bacteria, we would then inject them into the body. I have a feeling that might have side effects. But what we decided to do instead was, once we found such bacteria, we would identify the genetic basis for that capability, the genes and enzymes that they had that allowed them to break down these compounds. And then we would introduce those genes and enzymes into human cells, thereby augmenting the capacity of those cells to break things down. So here's the first step, and as you can see, this paper came out in 2008. It was actually the first paper that was published out of work funded by us. And it went very nicely. We found bacteria that did the job. What you're seeing here is what's called an enrichment culture, where we take a whole bunch of different bacterial strains and we give them this stuff, 7-ketocholesterol, and we don't give them anything else to live on. The idea here is that if they can break down 7-KC, then they can grow because they can extract energy from it, and if they can't, they can't. So most of these strains can't. They just sit there like lemons doing exactly nothing. But a couple of them are doing very fine. You've got a couple of strains there which after only 10 days have consumed the entire material. So that's great. Then we've got to, as I say, find the genes and enzymes that they're using, and that turned out not to be too hard. This is one way that we did it. It's called mass spectrometry. We essentially identify the breakdown products that the bacteria are creating in the process of breaking down the original substance, and that allows us to infer what enzymatic reaction is occurring and use bioinformatics to identify candidate genes. There are other approaches. We also used expression analysis to find which genes were being activated when we gave them the 7-KC and so on. All of these methods seem to work pretty well. Long and short of it was we had a few candidate genes by about 2010. That's when things started getting tricky. Step 3 in this process is the hard one, which is to actually get these enzymes to work in a human cell. The reason that's hard is because human cells are very different from bacteria. Bacteria don't even have lysosomes. But we managed it. First of all, we actually had to do the localization. We had to get our enzymes to go to the lysosome, and this shows that we could do that. The red on the left is a stain for the lysosome in cells. The middle one is our engineered protein, and the fact that they're overlapping reasonably well shows us that plenty of our enzyme is going to the right place, so that's all good. But at the end of the day, we also had to show that it worked, and that was what we were able to show maybe 18 months ago. This graph is a summary of the results. Essentially what it says is that if you give cells a completely insupportable amount of 7-KC on the right-hand end here, then it doesn't matter what you've done to them in terms of giving them extra enzymes, they die anyway. But if you give them a relatively modest amount of this toxic molecule, then the fact that the right-hand bar of each of these groups is higher than the other bars, that basically says that the cells which have the enzyme suitably targeted to the right part of the cell, the lysosome, are protected. They survive better than other negative control cells, cells that don't have an enzyme, or they have the wrong enzyme, or they have the enzyme not targeted to the right place. So this is an extremely good proof of concept. We're very happy about it. We have a couple more steps to go before we think we would be able to take this to the private sector. First of all, we would need to get this working in a different cell type. We did this in fibroblasts, which happen to be easy to work with, but we need to get it to work in macrophages. Then we need to move to mouse models of cardiovascular disease and show that it's protective there as well. But we're well on our way. I think I can probably trust you to understand that this really is a very effective proof of concept. And we're doing a lot more. We only have $4 million a year or thereabouts, but we are spending it very efficiently. We have a headquarters in Mountain View, California, just a couple of miles from the Googleplex, that has a few thousand square feet of lab space where we do 3 of our major projects. And we have more than a dozen other projects at various universities around the world. That one I just described to you happens at Rice University in Houston. We also have an educational arm, which I always like to mention because I think it's very important to understand that we're trying to grow the next generation of rejuvenation biotechnologists. So that's something that you can read about at the website, of course. Now, I thought I should mention a little bit about credibility, because maybe 10 years ago, some of you may have already come across me, it was actually pretty tough to sell this concept. I think you already got the idea from my early remarks that the whole maintenance approach is a big departure from what people had previously been thinking. The people who were working in gerontology viewed this whole idea of damage repair as a complete solution, as a very, very implausible idea. It was as far away from their historical thinking as the geriatric medicine approach was. So I had to do a lot of education, and it took a while. But the good news is that it really has been very effective. We are now in the very heartening position of having this kind of research advisory board of very obviously explicit endorsers of our approach. Every single one of these people are, really, really prestigious dignitaries in their various biological disciplines. And so there are obviously still a few people out there who just refuse to be educated about this, but by and large, the credibility barrier has been breached, which is very nice to know. But still, we've got to do more. And one thing we have to do, of course, is publish. We have to demonstrate the validity of what we're doing in the traditional manner. And that's, of course, what we're doing. So these are just a small selection of the papers that we've published recently. The one at the top is the one I've just mentioned, of course, the rescue of cells from 7-ketocholesterol. But we've done a whole bunch more than that. A couple of months ago, we had a lovely paper in a very prestigious journal, Journal of Biological Chemistry, showing that we could address senile cardiac amyloidosis using antibodies that would break the stuff down. A couple of other examples there. So things are going nicely, but of course, there's still a very long way to go. I'm pretty much done. I'm just going to spend a few minutes on other matters. First thing I'm going to tell you is what I'm not going to tell you. In other words, I don't really do the sociological considerations. I do have to. And some of you probably know that I do a brutal amount of media. I do probably 100 interviews a year. And the bulk of my time in those interviews is spent answering questions along the lines of, oh dear, where are we going to put all the people? Or won't dictators live forever? Or, won't we all get bored? Shall we say I have become a little bit bored myself of these questions. I regard them as being ultimately founded on a degree of myopia with regard to the importance of this work, a degree of ability to put out of one's mind the fact that we have quite a bad problem today with aging, namely a problem of 100,000 people every single day dying of it. That's 2/3 of all deaths. In the developed world, it's something like 90% of all deaths. And of course, it's not just the death; it's also all the suffering that most people have for a long period of time before they die of aging. So it's fairly obvious that this is not really the right way to think about it. We shouldn't be thinking about these problems. We should be thinking about this: that if we were actually to move to a post-aging world, we would have no age-related ill health. People's health would not be a function of how long ago they were born. The elderly would still be contributing wealth to society, so these therapies would pay for themselves. The elderly would not be a burden on their kids anymore. This is obviously something that a lot of the elderly worry about a great deal. So that's the kind of way I think. All right then, so final point, call to action. What can you guys do? As far as I'm concerned, this is really where I want to place emphasis, because whenever I give a talk like this or an interview, I feel I do get people on side. I do get people to understand that, yes, this is a really important problem, and yes, we do have a respectable chance to actually solve it. But I need to actually make sure that we solve it faster than we otherwise would. And at the end of the day, it all comes down to these things. First thing is, ultimately, it all costs money. 10 years ago, 15 years ago, when I started out, I had 3 problems to solve. I needed a plan for how to solve aging, and that's what I've been telling you today. I needed people. I needed the world's best experts in the various scientific areas to be enthusiastic about working on this, and that I have already, as I hope demonstrated, we already have now, very much. But at the end of the day, these people are still, by and large, sitting on their hands, waiting for the resources to actually get on with it, because we only have perhaps 1/10 of the budget that we would need to be able to fund everything that really needs to be funded. And that is absolutely the number one problem right now. Obviously, I'd like you to learn more. I've only had an hour. That's not nearly enough time to do justice to all that we do. Anyone who wants to get involved in terms of actually learning more about it through our educational initiative, please go and have a look at that at the website at sense.org. But ultimately, it's all about advocacy. Hands up, anyone here who doesn't know anyone who's richer than you are? All right. So it's all about changing people's minds, right? It's all about changing people's minds. The more people we have on side who understand the importance of this mission, the faster it's going to happen. And I spend a lot of my time getting people to understand how important this is. If the people that I were able to bring on side were to go out and bring other people on side, and so on and so forth, we would solve this problem a great deal faster. I wrote this book several years ago. It's very comprehensive. I call it semi-technical. What that basically means is you don't have to be a trained biologist to understand it, but you probably won't be reading it in one sitting. It's quite dense. It doesn't cut any corners at all on the science. And it was written several years ago, but it's still fairly up to date. That is not because there has been inadequate progress in the meantime. The progress has been wonderful. But what it is because the progress has been pretty much the type of progress that we predicted would happen. In other words, it is again another piece of evidence that the sense paradigm is standing the test of time. So that's all very heartening. I think I should also mention this. I don't know if any of you are going to be in California a few weeks from now, but I guess you probably have friends who are. And anyone that you might want to recommend to come to our conference, I would very much encourage you to do that. This conference is happening just maybe 10 miles south of the Googleplex in the August 21st to 23rd. And of course, it will cover all of the science of what we're doing of rejuvenation biotechnology, but also it will have extensive participation from industry, from regulatory people, from policymakers, and so on. So it's going to be quite a gathering, and of course lots of lay people there as well. And I very much encourage you to encourage other people to do that. I'll stop there, and I think there's probably a bit of time for questions. Thank you very much.
Hi. So in the field of cancer, we see that there are some cancers that have been have been tackled extremely well in the last 100 years, others that become very resistant and very difficult to deal with. Do you see the same things likely to happen when we tackle aging more generally, and that some diseases will be eradicated quickly and others will take a much longer time to get rid of in a long tail?
Yeah, that's a really great question. So, first of all, let me give you my take on where cancer sits within the whole universe of problems of aging. My view is that cancer is by far the hardest aspect of aging to fix. It's the one that has natural selection on its side, and that makes it, any cancer that's big enough to be clinically relevant has a trillion different individual engines of genetic creativity in it. It's really a tough nut to crack. Now, a lot of people, because of largely what the kind of evidence that you described, they don't like to think of cancer as a single disease anymore. They like to think of it as a family of diseases that we just happen to give some collective name to. But the fact is that's not really fair, because there are some things that cancers have in common. And in particular, there's one thing that absolutely all cancers have in common, namely the fact that they need to figure out a way to grow the ends of their chromosomes. Chromosomes get shorter when a cell divides, and there needs to be a way to compensate for that. There's only two ways that cancers ever use to do that, and we're well on our way to being able to stop that from happening. So that's essentially the way that we're going about it. In general, when you talk about the relative degrees of difficulties of coping with aspects of aging, I think you're right. There are definitely things that are harder than others. And actually, a large part of the reason, well, in fact, most of the reason why Sense Research Foundation was set up was precisely to mitigate that problem, to ensure that we work on the hardest things and that they don't get left behind. And, we just fix the easy things and end up where we were.
Thank you.
If you had like a crystal ball, how long do you think it will take until you've got something that you say actually will be able to live for another 100 years?
Yeah, you asked that question in exactly the way that an engineer should ask it. In other words, I haven't the faintest fucking clue because it's a long way away. But, of course, you want an estimate. Right. So and actually, you're quite right to ask the question, because a lot of gerontologists would just refuse to answer. They would say it's irresponsible to get people's hopes up or something like that. And I say, on the contrary, it's irresponsible not to give an estimate because, however bad my estimate's going to be, yours is going to be even worse, right? And therefore, I've got to give give as good an estimate as I can. So my estimate is this. I think we have at least a 50-50 chance of getting the technologies that I described today working reasonably well within the next 20 to 25 years.
Yes.
Right? And the technologies that I described today, I think, will probably give about 30 additional years of healthy life. And of course, they will give those years to people who are already in middle age or older, 60, 70, or even maybe 80, at the time that the therapies arrive. So that means most people in this room have a relatively good chance of benefiting. Two caveats. First caveat, everything I just said is only going to be true if research in the next 5 or 10 years goes a lot faster than it's currently going. The funding limitation that exists at the moment, I estimate, is slowing things down by roughly a factor of 3. In other words, over the past 10 years, the amount of progress we've made towards getting proof of concept of these things in the lab and so on has probably only advanced by about 3 years relative to how well it could have gone, if only science had been limiting. The second caveat is the probabilities. So I can tell you a 50-50 estimate, but I want to tell you right now the variance is high. I think there's at least a 10% chance that we won't get there for 100 years. But who cares? A 50% chance is perfectly enough to be worth fighting for.
Yeah, thank you.
Thank you very much for this very interesting talk.
My pleasure.
Actually, about a year ago, we set up this company called Calico, the California Life Company. We set it up to work independently. And I was just wondering if you, first of all, how do you view this development? And secondly, if you've already had the chance to work with them so far?
Yeah, actually, I was very surprised that no one at the talk I gave at lunchtime asked about Calico. But yes, Calico was, as you say, set up by Larry and Sergey about a year ago. And, my view of that development was expressed in Time magazine, which was where the thing was announced. Time got me to write the reaction piece, so to speak, and I was pretty effusive. I was overjoyed, and I still am. I think there is a very respectable chance that they will make a really big difference. But only a chance. It just remains to be seen. They are taking their own really good time to decide exactly where they're going to be prioritizing their efforts. And in a way, that's a good thing. The worst thing that could possibly happen would have been a repeat of what happened 15 years ago when another very wealthy individual, Larry Ellison, decided to do essentially the same thing, albeit in the foundation rather than the company. he basically went out and promptly hired a veteran gerontologist from the NIA, and the result was completely predictable. 15 years and $400 million later, absolutely nothing had been done that wouldn't have been done anyway. So it was, and he's given up now. So Calico is potentially a really good thing. They've got people at the top who are, by and large, not trained gerontologists, and therefore they know what they don't know. They're making their own decisions. They're not talking to us nearly as much as we'd like them to, and as we think they should be, but that may change. So I'm hopeful.
Thank you. Hey, Aubrey. If, as you describe, this might become one of the biggest industries ever, and
the biggest.
And if, as you describe, you've already done a great job at convincing people that it's worthwhile fighting for it, what are, in your opinion, the 3 main obstacles to actually get the necessary funding going, which seems to be one of the biggest problems on the way?
Yeah, I would actually just stick with one main obstacle, which is something that I historically used to call the pro-aging trance, but my marketing people tell me that that's too derogatory, so I'm not allowed to say that anymore. Essentially, it's fear. It's a combination of 2 types of fear. Number 1, fear of the unknown, recognition that a post-aging world will be very, 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 very different, and therefore, maybe we'll stick to what we know. Kind of essentially the same as what happened to resistance to the Industrial Revolution. And secondly, fear of getting their hopes up. People just are not willing to re-engage this battle. People in general have made their peace with aging. They have accepted defeat, and when you've accepted defeat, the last thing you want to do is go through all that stress again. So that's basically what it's all about. People who are otherwise perfectly well educated and perfectly rational in absolutely every other walk of life will say things and think things that they would be embarrassed to say to a small child to shut them up when it comes to this particular topic. And of course, the problem is that I don't get to talk to everybody. Time and time again, I will be in the company of people who not only get it, but say they get it. And they will tell other people that they get it and that things are good. But do they actually write me a check? Not as such. Why do they not write me a check? Probably because their wife doesn't want them to.
Hi. The cost of gene sequencing has decreased kind of massively over the last 10 years, and I've seen various predictions, people saying that in 5 years' time we'll all be getting our gene sequenced for the cost of $10 or whatever. Do you think it's a high chance that we might find genetic causes for a lot of these problems like Alzheimer's before we find solutions that are involved with damage limitation?
Yeah, great question. So I'm going to answer it in a slightly complicated way. I'm going with 2 definitions of the word solution. There will be the possibility of identifying genetic factors that are responsible for the age of onset of certain age-related conditions, whether it be Alzheimer's or anything else. And then there is also the possibility of genetic conditions that are simply responsible for it in a binary way, for it happening or not happening. So what I can tell you is that it's very likely that we will find the former, albeit it's not nearly as easy as people were thinking maybe 20 years ago when apolipoprotein E was discovered. And it looks like it will involve a lot of different genes combining in different people, each individual one of which only makes a small contribution. However, we can say pretty definitively at this point that the answer to the latter question is no, that the aspects of our metabolism that ultimately drive the accumulation of these various types of damage and therefore the eventual arrival of these pathologies are non-negotiable. No, the number one worst thing that we have that drives all these things is breathing. Breathing is really bad for you, but there's not a lot of choice that we have about it.
Cool, thanks.
Hi, we've talked a lot about sort of funding and where you get that from, and also bringing in the private markets also narrows the diversity of the research. So, at what stage do you go to the private markets and the venture capitalists?
Yeah, great question. I'm not really sure about narrowing of diversity. I mean, in a sense, the continued existence of non-profit participation in all of this is a good protection against that. So, individual components of the range of things that need to be done might go into the private sector without necessarily restricting the ability of other components to continue being developed, in the philanthropic sector, so to speak. That's kind of how I see it going. I think we may be done. Well.
Hi. This may not be of interest to you at all, but sticking with your car analogy, if you take a car and you drive it on a smooth road and you keep it away from sand and you put in the right stuff that it needs in the engine and water and everything, it probably accumulates less damage. If we look at that for a human being, in the meantime, before your research is going to save us all, what can we be doing to damage limit?
That's an absolutely excellent question and an absolutely excellent way of expressing the question, because you're absolutely right. In the case of simple man-made machines like cars, lifestyle matters a lot. You can drive a car carefully and well, and it'll last a lot longer, quite a lot longer than if you don't. The unfortunate fact is that it's not quite like that for the human body. When I mentioned a moment ago that breathing is really bad for you, breathing and the other non-negotiable aspects of being alive, like eating, or eating anything, not just eating badly, are the overwhelming majority of the drivers of the accumulation of damage. Of course, there are things that you can do that are obviously bad for you, like smoking or getting seriously overweight, but nothing your mother didn't tell you. All right. If you're basically averagely healthy, then the amount of additional postponement of age-related ill health that you can achieve by any kind of lifestyle or diet or exercise modifications or whatever is, as far as we can tell, really tiny. And therefore, there's only one way out of this, which is to hasten the development of therapies that we don't yet have. In other words, give me large amounts of money. Thanks very much, everybody.